AIDRM, A.I.D.R.M. Good abbreviation? Maybe so. Reading scientific articles may not be a walk in the park and instead feels like a long bus ride. You can start losing sight of the purpose of reading and forget what you're looking for. An article is essentially divided into 5 sections, and it's best read in the order abstract, introduction, discussion, results, and method. Each section has different aims and goals, so following along the checkpoints will help orient readers to the right destination. Here are some prompt questions you should be able to answer and highlight after completing each section of a research article. Number 1. The abstract. This is the first short preview paragraph you will see in any research paper. It provides readers a general summary of what the article entails. In this section, you should be able to identify the main questions the researchers are trying to answer, the goal of the research, and what the researchers want to achieve. In addition, a vague statement of what the research findings concluded. The sign of a well-constructed abstract is the relevance of their study. Why and how does it apply to me? Now let's explore the example article I've chosen for today, called Non-Invasive Early Detection of Cancer 4 Years Before Conventional Diagnoses Using Blood Tests. The goal of this article was to prove that the researchers' new technology called pan-SEER was valid and impactful for detecting asymptomatic cancer. The application was pan-SEER technology can be used to detect asymptomatic cancer or cancer at early stages, and in turn hopefully increase the chances of successful treatments and cancer survival rate. Number 2. The introduction. An introduction is the support system of a paper. It works to provide background information. It includes many references to external research findings. It's essentially a reflection of the researcher's thought process. You should be able to answer the questions, what we already learned from other studies, what are the holes and unknowns of the specific topic, and if applicable, try to identify any current innovations, treatments, and applications of this knowledge. Generally, the introduction shouldn't take too much of your mind space. Casually read it, and if you're curious, do some external research for points of confusion. In the article, the questions are answered as the subject of the study is cancer, the current knowledge is that cancer can be detected by screening for ctDNA, which is circulating tumor DNA. The unknowns of this field is there are difficulties in detecting earlier stages of cancer because people in earlier stages of cancer have smaller amounts of ctDNA, which is usually not detected by alternative methods of cancer screening. Of course, the research goal will always be restated in the introduction. Now switching over to line 3, it's the discussions. Discussion is the interpretation of the results. There's no figures or data included, but it's where all the conclusions are drawn. You should be able to restate the goal of the research, the conclusions of each figure, how did the results contribute to the main research question, how was the interpretations of the result change or enhance our current understanding of the field. It's also encouraged to do some brainstorming, come up with some future experimentation suggestions. Anything you wanted them to test out, but they didn't. If something in the discussion section does not make sense to you, then always write it down. If the jump to the conclusion was too big, or if the data was unclear, asking questions does not mean you are incapable of reading, but instead is a reflection of your understanding. In the article's discussion, they concluded that pan-SEER had significant success rates in the detection of asymptomatic cancer using methylation markers. pan-SEER is a fast and less invasive, cheaper alternative cancer screening method, which is perfect for clinical settings. Uniquely, you will also find that the article will disclose limitations of their study in the discussion section. The results section: diagrams, data, and figures. Here, the researchers briefly explain each experiment and the data they've chosen to collect and analyze. Keyword is chosen. Everything researchers include is strategic, and sometimes figures, internal or external, may be excluded from the results section. Excluded figures can be found in the supplementary figure section. Most times it's not necessary to look at supplementary figures, but you can make that decision. The goal after reading this section is to create one sentence that includes the purpose and conclusion of each data set, and to note down any areas of confusions. At the end, pick 2 top figures that present the most significant results. For this article, I chose figure 2 and highlighted how the researchers proved that high levels of accurate cancer screening for various types of cancer was achieved through the use of pan-SEER. The final section is the methods section. It should provide enough detail for all the experiments to be replicated in other labs. You should be able to generally outline the chemicals, environmental conditions, cell lines, and experimental procedures and groups that were used during the research. Then set some time aside to Google search and answer questions like, what are the chemical mechanisms? What part of the cells do they affect? The type of cell lines that were used. What is the experimental technique used for? List your findings and keep it as a reference when revisiting the results section. Google is essentially your best friend in this section, so take your time, but avoid digging a rabbit hole. Get the information you need and then stop. Don't overwhelm yourself. Some recommendations for reading articles are using applications like PDF Markup, GoodNotes, or simply Google Doc to annotate, highlight, and organize the information of the articles. There are different types of reading, deep versus just the type to get the literature values you need for a chemistry lab report. Whatever type of reading you're doing, each person has their own preference in reading order. This map is meant to be a prompt or an inspo for developing your own map. And furthermore, each subject can have a different order for effective reading. And that's why I've left an empty template for you to fill out. Just keep practicing and find your own rhythm. I hope this subway pathway can be your gate into the research and science world. As always, you can screenshot any of the posters that were in the video. And that's all for today. Keep translating science.